Hey everyone, my name is Jihoon Ryu, and my English name is Chris, and I'm from a Daegu International School, which is an international school of an American curriculum located in Daegu, South Korea. And my project is named Exploring the Role of Non-Coding Strand, Antisense Transcription, and Gene Regulation. First, I'll present my rationale. I had two main purposes: to test the trend of antisense transcription and compare it with the features of the sense transcriptions, and the 5 features are listed below. And my second purpose was to explore its importance and possible applications in the future. And these two purposes became one research question. And the research question was, how do the general trends of antisense transcription compare to those of sense transcription, and what are the potential future application of these findings? So before moving on to the results section, I would first like to introduce the mechanisms and the foundations. Antisense transcription was first discovered by Buckrinsky MI, and he discovered NATs in early stages of HIV infection. The letter in the middle shows his discovery of the letter to a research company. And then the picture on the right shows how he found out about the antisense transcription and the plus strand. So first, NATs were regarded non-functional. Researchers, when finding out about NATs, they thought that NATs were same as junk DNAs and not posing any other function except for just degrading. That is because they had low expression levels, unknown functions, and abnormal heterogeneity. However, recent developments such as the third generation DNA sequencing developed by Stephen Quake enabled a deeper analysis of these discoveries. The images on the left show how NATs were regarded as junk DNA due to these reasons. Now, NAT's abundance led the researchers to think about and research and delve deeper into the topic of antisense transcription and non-coding strand because of how abundant they are. There are a very large number of NATs that exist in the body, in human body, but they do not encode for proteins. That was what made the question very weird, because if there are a lot, then why would they not code for proteins? And to delve deeper into the statistics, complex eukaryotes such as fungi, plants, and animals have more than 50% of NATs, and nearly 99% in human genome have NATs. And scientists started to realize that non-coding RNA or NATs does play a role. So these questions came up into my mind, and so prior research, I decided to delve deeper into this research topic, and this is my methodology. These are the 4 main stages of my progression. So first, I obtained the genome-wide samples from samples of yeast, and that's the scientific name of yeast. And then I refined those data into meta-array. And meta-array simply means data that consists of all different types of information into one single array. And then I extracted genes with high and low antisense transcription to obtain the data and analyze them compared to their antisense transcription levels. And at last, I did the calculations and the analysis and wrote it into one finalized coherent research paper. These are my following code sequences, but instead of delving too deep into every picture, these are the main simple 3 stages, which is the first image shows how I produce the meta gene array. The second is how I extract the genes with high and low antisense transcription. And the last was how I calculated the stability levels for extra analysis. And now delving into the main topic, which are the results. The general trend of antisense transcription is shown in the bar plot below. There are genes with high transcription and low antisense transcription, which are shown as the height of each bar. And you can see that the bar heights are relatively similar to each other. That means that there's an even occurrence of antisense transcription among the gene. So if there were too much of either one, then that means that antisense transcription is just a bizarre thing that happens in the genomics. But however, since there are even amounts of high antisense transcripted genes and low antisense transcripted gene, we could analyze that this process happens fairly evenly among the human genome and also other animals too. As well, the x-axis represents the position relative to TSS, and TSS means transcription start site, and this is based on the sense transcripts. So if there are two strands of the DNA, the TSS is where the RNA polymerase attaches to the main strand where it becomes a protein. And comparing the sense transcription and antisense transcription levels, you could see that the blue line reaches the peak when it comes to TSS, and that is what we want to see because the gene starts producing a transcript starting from the TSS, and that's why we could see that the sense transcription levels are so high. But however, the interesting thing is that antisense transcription drops, and it has a negative correlation with sense transcription in some regions. And simply said, that means that if sense transcription show a concave down curve, then that means that antisense transcription shows a concave up curve. This is a simple explanation of how antisense transcription works. Again, the TSS is based on the blue arrow, not the red arrow. Then analyze the nucleosome levels and antisense transcription. This was obtained to MNA-seq level, which is a microcochleal nuclease sequencing dataset. And we could see that the blue line in the graph drops way severely than the red curve in the graph. And instead of just explaining in words, I'll show you a simple animation. So you can see that blue lines have a more severe peak, and the blue line represents genes with high antisense transcription. And if there's a lower, that means that there's a lesser chance that a nucleosome will occupy there. Then that means that if there's a lesser chance of nucleosome occupying there, then that means that RNA polymerase II will easily bind to the gene. And then RNA polymerase II will easily transcribe and produce transcripts. So that means that if there's high antisense transcription, then it correlates where it's a higher chance of RNAP2 binding to the gene. However, with low antisense transcription, there's a higher possibility of a nucleosome being there. That means RNAP2 can't bind to the DNA. Therefore, we can't really produce a transcript that will eventually become a protein, and all that process will be gone. So that is an interesting mechanism to see. As well, I compared transcription factor levels and antisense transcription. Again, this plot could be easily shown by an animation, and you can see in plots A, B, D, and E, and I'll explain why I excluded figure C. But however, in A, B, D, E, we could see a peak. We could see a higher peak in blue line than the red line. And again, blue line represents high antisense transcription, and red line represents the opposite. And in the blue line, if there's a higher peak in the high antisense transcription, then it correlates with a higher level of that transcription factor being attached to the RNA polymerase II. And these transcription factors are known to help the transcription process. So again, with the animation, if the RNAP2 binds to the gene, then there's a higher, if the gene has a higher antisense transcription, then it correlates with a higher chance of these transcription factors attaching to the RNAP2 and some of the proteins attached to the TSS, and then these transcription factors could assist the transcription process throughout the process and then produce an NAT with a higher success rate. Next is histone modification levels. There are a lot of different types of histone modifications, and the one specific type I delved deeper to is H3K4ME3, which is trimethylation of lysine 4 on histone H3 protein levels. Again, the blue and the red line represent the same things, and you can see that behind the TSS and near the end site of the gene, you can see that genes with high antisense transcription have higher levels of H3K4ME3 levels. And again, this data was yielded by the ChIP-seq datasets. And that means that if genes with high antisense transcription have higher H3K4ME3 levels, then it will foster the transcription process. And this was written in the research paper by X. Huang et al. In 2019. And the histone modification generally assists the build of transcription machinery and mediates gene induction. So we could say that high antisense transcription correlates with higher H3K4ME3 levels, which fosters the process of transcription. The last result of my research paper was about stability. And the x-axis represents the stability level, and it ranges from 0 to 100. I scaled it like that. And the y-axis means the gene count, which is the amount of gene that has a certain type of stability level. As you can see in the bar chart of A, which is sense transcript stability levels, there are more than 3,500 sense transcripts that have nearly zero of the stability level. That means that a lot of sense transcripts have low stability levels. And in the figure B, you can see that only less than 1,500 natural antisense transcripts have a stability level of zero, but however, it shows a less severe drop. That means that there are more antisense transcripts that have a stability level of 100 than the sense transcript that have a stability level of 100, and you can see by putting it side by side. So that means that NATs have less extreme stability levels and are evenly spread. But however, this may have been produced by inefficient equation, because the only thing I did is to divide transcription levels over the levels of transcripts. So that means that there are way more sense transcription levels, as you can see in this plot again. There were way more sense transcription levels. So that means that it increased the denominator. So when dividing the two dividend, and when dividing the denominator and denominator, the denominator increased so much that it caused the sense transcript levels to become nearly zero. So that might have been the flaws of this calculation, since most of the research papers say that NATs have a low amount of stability. And now the discussions of the results. So first is, did I explore convergent antisense transcription or PAS-associated antisense transcription? And the answer is I looked at both, because I looked at throughout the gene, I looked at the antisense transcription levels throughout the gene, and also before the TSS start. So as you can see, the TSS, convergent antisense transcription, goes over the TSS, and we can see that our the red line in the middle plot extends beyond the TSS as well. And also, the graph on the right, produced by Kingren et al. In 2020, showed a drop compared to sense transcription levels. And you can see similar rates because a concave up curve in the antisense transcription, and you can see that this result is somehow similar to Kingren et al.'s research paper and is similar to different literatures that are already published. As well, Dr. Strand Murray was one of my mentors, and I compared my results to his work as well, since he was the one who suggested this research topic with me. And the thing is that in level A, you can see that genes with high antisense transcription show the higher H3K4ME3 rates behind the TSS site, and you can see that that's how it fosters the transcription process. And in B, you can see that the two transcription factors that are same with me, the TBP and TF2B, were also showing a higher peak with genes with high antisense transcription. And as well, in C, you can see that the nucleosome occupancy drops severely to help the RNA polymerase II to bind to the gene and foster the transcription process. And again, why did I exclude that C figure in the transcription process? So one research I did was that the RAB1 protein assists transcription, and that is what I knew in the past. But however, through more researching, I also figured out that RB1 protein also blocks transcription. So that may explain why RB1 has a higher peak of red, which is low antisense gene transcription, than the blue line, because it helps block transcription, which hinders the sense transcription process. So we could see that RB1 has a negative correlation with transcription. The overview of my research was that, first of all, I delved into the non-coding strand, which codes for the antisense transcription, and through processes of 2, 3, 4, to ChIP-seq, transcription factor level, also ChIP-seq, and nucleosome level, MNA-seq. Through analyzation of those data sets, I could finally figure out that the non-coding strand, through mediating processes, can impact gene regulation, which correlates with gene regulation. And then here's another example of how antisense transcription regulates the gene expression. So this is transcriptional interference, and when two RNA-P2s collide with each other, it hinders the process of transcription, and that is how it hinders with the gene expression process as well. So here's a easy animation of you to understand. And also antisense regulation, when these two produces NAT, which is red, and sense transcript, which is blue. These two transcripts bind to each other, forming a double-stranded RNA, also known as dsRNA. And then a nucleus comes into the dsRNA, therefore degrading the dsRNA, and therefore the RNA cannot become a protein. And that also hinders with the process of gene expression because it entirely blocks the transcription into becoming a translation process. And all these processes interact with each other. This is the main process I found also in my research paper. So A, B, C, as you can see in this figure produced by Brodoi and Chatterjee in 2015, you can see that all these processes interact with each other, and therefore you could conclude that non-coding strand is directly correlates with gene expression. And in conclusion, non-coding strand through meeting processes contributes to gene regulation. And here are the application, which is my second purpose. So ASO, antisense oligonucleotides, are accepted by the U.S. Food and Drug Administration to treat these two dystrophies, which is Duchenne muscular dystrophy and spinal muscular atrophy. So we could see that the discovery of antisense transcription would lead to any other applications in the medical and the biological field, and is impacting the world currently as well. So thank you for listening to my speech, keynote slide, and please contact for inquiries.
Thank you, Jihoon, for that very thorough and well thought out presentation and for sharing your research with us today. Greatly appreciate it.